In this video, I will be critically appraising a research article. Now, I have used certain set of questions that was sent to me by my supervisor, but you have the freedom to use any sort of questions depending on the article type that you have. For instance, this link that I will be posting in the description provides you different checklists for whatever type of study that you are trying to critically appraise. For instance, this is the CASP checklist. If you use this link, you will find certain questions with yes, can't tell, and no, and you will go on this way. You will be able to determine whether or not the article is worth continue reading, and you will also be able to judge on the basis of these questions on whether or not this article is strong in evidence or weak. In this example, I will be critically appraising a research article. The components of this video will consist of an article summary, critical appraisal of the introduction, followed by the methodology, followed by the results and discussion, and lastly, we will also score our article on whether or not it's weak, moderate, or strong in evidence. The article that I have selected is titled Scintigraphic Patterns of NDM-111 Oxine-Labeled White Blood Cell Imaging of Gram-Negative versus Gram-Positive Vertebral Osteomyelitis. For those of you who don't have a radiopharmaceutical background, the NDM-111 oxine is a radiopharmaceutical that is attached to the white blood cells in order to help us image where is the accumulation of these white blood cells happening. Now, it's a mouthful, I do realize that, but once you get to the procedures and what they were doing in this article, I believe you will find it very interesting. If we look at the experimental part of this article, we are trying to induce vertebral osteomyelitis in the rabbits by either gram-positive bacteria or gram-negative bacteria. We divide the rabbits into two groups, each group containing 25 rabbits, and out of the 25, only 16 successfully infected rabbits were used. These 16 rabbits were infected differently, where 8 of them were having induced vertebral infections and the other 8 having femoral infections. Following the infection, we inject these rabbits with NDM-labeled white blood cells for the purpose of imaging. We take in the observations after 24 hours and we realize there is a major difference between the first group and the second group. And that difference is that the first group, whether it's a vertebral infected rabbit or a femoral infected rabbit, the groups showed high accumulation of the NDM-labeled white blood cells. However, when we compare it to the second group with the gram-negative bacteria, there is little to no accumulation in the majority of the rabbits. Why is this happening? The reason why this happens is attributed to the gram-negative bacteria E. coli's ability to produce anti-chemotactic factors. Now, these anti-chemotactic factors will prevent the accumulation of white blood cells in the site of infection, therefore having little to no accumulation rate. So this is the hypothesis question that the investigators are trying to prove. Is gram-negative bacteria secreting anti-chemotactic factors the reason behind the non-accumulation pattern of NDM-labeled white blood cells? This is the part where we start our critical appraisal. So we begin with the introduction. So this is the article. As we mentioned before, we are using NDM-11 oxyquinolone, where the radioisotope that is responsible for the imaging is the NDM-111, and the pharmaceutical particle is the oxyquinolone or oxine. Our first question regarding the introduction: is the article or the research question relevant to the assignment? Now, our assignment was to select an article with a radiopharmaceutical. So yes. Is the journal peer-reviewed? Now, the journal of this article is Medical Principles and Practice. Looking into the quality of the journal, it's a Q2 journal. It has been since 2017, which was the year of the article publication. And looking into their website, we realized that there is a double-blind peer review system where the reviewers don't know the names of the authors and the authors don't know who reviewed their manuscript. Is the article recent or is it seminal? Now, it's a 2017 article as mentioned, so it's definitely a recent article. Was the hypothesis or the research question clearly stated, and was there a clear statement of what the investigators expect the study to find, which can be tested, confirmed, or refuted? Now, for these two questions, the answer can be found in the objective statement, where we can see that the goal of the study was to investigate whether or not gram-negative organisms that secrete anti-chemotactic factors cause the non-accumulation pattern of indium oxine-labeled white blood cells. We can also see from the objective statement that this study was used to investigate whether gram-positive or gram-negative bacteria infections cause the uptake of indium-labeled white blood cells at the site of osteomyelitis in the vertebra or the thigh bone. Next is to critically appraise the methodology. Was the study design appropriate for the research question? Yes, because they used the radionuclide imaging or in vivo imaging, which is a non-invasive visualization of the living organisms for research or diagnostic purposes. Did the methodology address important potential sources of bias? The answer is no, because we have different types of biases. We have selection bias, measurement bias. Now, if we have a randomization in an article, this reduces the confounding variables. And if we have a blinded randomization, then the blinding reduces the detection bias, both of which randomization and blinding were not carried out in this research. However, a third type of bias, which is the attrition bias, was actually reduced because the research did include the reporting of animals excluded. However, because the other types of biases were not detected, we say that the methodology did not address all the important sources of bias. Were both inclusion and exclusion criteria described? The answer is yes. So we can see in the article that the methods of inclusion supported that there were two groups used, each group containing 25 rabbits. Out of the 25, 16 successfully infected and surviving rabbits from each group were used for the imaging and the analysis. And of the 16 rabbits, each group included 8 with vertebral infection and 8 with femoral infection. While on the other hand, the exclusion criteria was also mentioned that the 18 rabbits were not included in the study due to unsuccessful induction of infection, death, or other technical difficulties. Were the statistical tests appropriate for the study design? Now this is where it gets tricky, because this particular study did not have statistical test reporting. And this is a big flaw in the study, because pre-specifying endpoints and analysis, recognizing the common underlying assumptions of statistically independent observations, performing whether a sample size calculation or addressing the multiplicity issues are very important parts of an in vivo study. Was the sample size appropriate? Again, this information was not available in this article. There were no sample power calculations. The sample size is very important to establish the power and the impact of the study. So the determined size should be optimum and must be obtained by the scientific method. The arbitrary calculation with less or more can affect the study design and its significance. Was the data collected in a way that addressed the research question? Yes. The figures in the article did show that gram-positive bacteria showed higher accumulation of the indium-labeled white blood cells at the site of infection. As we can see here, there is a big lump of light compared to the gram-negative bacteria, which showed either very minimal accumulation or no accumulation. Did the research methods limit the influence of confounding variables? No, because the only way to limit the confounding variables effect is to do randomization, which was not done in this article. Have ethical issues been taken into consideration? Yes, we used rabbits, and we can see as part of the article, experiments were performed in accordance with guidelines approved by the Institutional Animal Care and Use Committee of Kuwait University. Lastly, we will be appraising the results and the discussion. So I put this part as a Q&A section. Was the study performed according to the study methodology? Yes, as we mentioned before. Were the statistical analysis performed correctly? We didn't have any statistical analysis. Is there evidence of statistical fishing, like changing statistical tests to ensure significance? Again, we didn't have statistical analysis to begin with to see whether or not they were fishing. Was there a deviation from the planned protocol? No. More Q&A. Did the results reliably support or refute the claims with a high quality evidence? Well, the results did support the claims, but the quality of evidence is somewhat questionable because there were no statistical data. Were tables and figures appropriately used? Yes, there were representative figures. Was the original data presented clearly so that the readers can check the statistical significance of the results? Again, data not available. In order to achieve the statistical significance, you need to do a t-test. For this article, if I want to suggest an improvement tactic, is to provide a t-test for two independable variables where we will be able to estimate the variance, we will be able to estimate the mean difference between the two groups, and we will be able to calculate the p-value. Do the results justify the conclusions? Yes, and as we can see in the conclusion of the article, it mentions that the majority of gram-positive infected rabbits were actually showing high accumulation of the indium-labeled white blood cells. However, in most of the gram-negative infected rabbits, minimal or no accumulation was seen. Is there a clear statement of findings? Yes. In the discussion part, we can see that the current study showed that the majority of infections caused by gram-negative organisms were negative, while the majority of gram-positive infections were positive in the indium-labeled white blood cells. Is there adequate discussion of the evidence, both for or against the researchers' arguments? Earlier, we did discuss the evidence that stood with the hypothesis. However, we did have certain deviations that included having a positive finding in the case with gram-negative vertebra and femoral infections that could probably be attributed to the degree of the severity of infections. Similarly, the negative finding of one case with a gram-positive organism could again be due to milder infections. So these are the claims, or these are the justifications for having deviated results from the actual hypothesis. And this is an example of a figure of a gram-negative bacteria that showed high level of accumulation of indium-labeled white blood cells at the site of infection. Are the findings discussed in relation to the original research question? Yes. What are the strengths and the weaknesses of the research article? So the strengths of this article was that it stated a clear objective. The design was also clear, figures were supportive of the hypothesis, and those that weren't were also presented. Data that deviated were justified. The weaknesses, however, is that there were no statistical tests. More Q&As. Assess the usefulness and the clinical applicability of the study. The study is supported by reason, but can't be fully proven without statistics. Does the study add anything new with evidence to the field of study? Yes, it did explain the reason behind the cold patterns resulting from gram-negative vertebral osteomyelitis. And do the authors have any potential conflicts of interest, and have these been declared? Conflicts of interest were not mentioned in the article. One might assume that they don't, but a clear claim should be provided. Who has funded the study, and can we trust their objectivity? The funding source was not mentioned either, so we can't tell for sure. And did the authors include relevant and important references? The answer is yes, and here are some of the references they used. Overall, if we take the answers to the previous questions into consideration and calculate the ratios of supportive answers versus the unsupportive, we will find that 61.67% were actually quite strong, while the rest was weak. This places the article at a moderate degree of evidence strength. For those of you who are curious about how I did the calculations, it's a subjective criticism, so it did not have any scores on its own. So I had to calculate the questions. There were 30 questions, therefore 3.33 points per each question. So I divided the overall range out of 100% so that 0 to 33% would be a weak evidence, 34 to 66% would be a moderate evidence, and 67 to 100% would be a very strong evidence. This article scores 18.5 for the yes, for the strength, and 11.5 for the no, or for the weaknesses. Therefore, if I want to evaluate the supportive evidence, I will take the 18.5, multiply it by 100, divided by the 30 questions, and it will give me 61.67%, which lies in the moderate region. Therefore, our evidence is of moderate strength. That was it for the critical appraisal. Hope this video was useful. Thank you for watching and hope you have a great day.